Bismillahirrahmanirrahim. Assalamu alaikum, dear listeners. Now we are going to perform step number 6 of our differential gene expression pipeline. So in this step we are going to measure the differential expression of our two breeds in a pairwise manner. So this is the step. You might have remembered that in step number 4 we have generated 3 files. So those files are present in the utils folder. This is the folder where our 3 basic files are over there. So we are going to use, let's copy the required file into our next folder, that is analysis. So I'm going to copy one of the file that we are going to use as an input file from util folder into analysis folder. So let's go into util folder first, cd util auto completion ls-lh. So here is the file, this is the file that I want to move. Okay, so I want to move this file. So move this file, copy it, move this file into one step back. There is a folder name analysis, I think, autocomplete. Okay, so we move it by other way, so it would not be confusion. So go into the folder section, into the trash bin. Okay, no, sorry, in our hard drive. So here we are, then we are in Trinity, then in Trinity there is a util, and here is the file, this file, and this file we want to move. So just right click here, move to here, analysis, and in here in differential expression folder. Okay, just select it here. So let's go back, and we will go to that folder on terminal. So definitely we have to come back one step back, cd dot. So we are in Trinity, ls-lh, we are in a Trinity, and we have to go to the analysis folder now, where we have just copied that input file. So cd analysis. So inside the analysis there is a folder named differential expression, so we have to go into the differential expression folder. So let's see, the file should be here that we have just moved, and that is with the name of yes, so this is the file Burberry Pashmina isoform.matrix. Okay, so first step, as you have seen it, that we have to move our input folder, input file that was in a util folder, and we moved it into the analysis folder, and then within the analysis there is a folder differential expression. So we just copy it because the command we are going to execute here, that is present in this folder. So our command is again a Perl command. So let me give you that command that is starting from Perl, because that is a Perl script. Okay, I have ran it before, so So not this one, not this one, not this one. Okay, so this is the command. So this is the Perth script. So I have to write the Perth, and there is in the same folder here, in the same folder there is a script named run_de_analysis.perth, and then we have to give the argument minus matrix, and this is our input file, and that should be present in our working folder. So that is the file that we have just moved in this folder. So the Burberry _pashmina dot isoform count dot matrix, this is the input file, and then we are using the method or the bioconductor package, that is edgeR. So we have to mention minus method edgeR, and then we are going to use minus dispersion 0.1, and this might be from 0.1 to 0.4, and the dispersion meaning that if you don't have the biological replicants, so you are giving the argument that you don't have the biological replicates, so it will automatically adjust. So before running this command, I have to install edgeR software, edgeR package on my R, because you know that Trinity is working on a R language, and we downloaded and installed the R. Might be I have already told you in a previous steps that we need edgeR in our analysis method in the Trinity. So we have to install edgeR. Otherwise you can also use DESEQ2. DESEQ2 is another R and bioconductor package for the analysis of the differential expression analysis of the RNA-Seq data. So there are a few other as well, but we are going to use the edgeR. So first we have to edgeR. So if we want to go to the R, open the R. Okay, so here we must have R, otherwise we have to install the edgeR. The package name is edgeR. Okay, so we have edgeR, so we can just refresh it. Okay, if you don't have the edgeR, you can install it from here. I think it should be here. Okay, if it is not here, you can install it from the bioconductor website. So you will go to the bioconductor. Let me give you an overview again here. So you will go to the bioconductor website, and you will just write the edgeR in the search, edgeR. So edgeR is here, so just click here and just copy this R script, copy it, go into R and just right click here and paste it here, and when you just enter it, it will start installing the edgeR package in your R. So in our Linux commands we are using Trinity, and Trinity is actually using the R script, and R need edgeR package. So this is the whole cycle and the whole scenario behind that. We are using Linux, but actually we are using the software Trinity, and the Trinity is running on R, and R requires the differential expression package and RNA-Seq analysis package called edgeR. So this is why we have to install the edgeR on R, and then we will go back to the terminal. So here once again we are in Trinity folder. Within the Trinity folder we are in analysis folder, and within analysis folder we are in a differential expression folder, and within expression folder we are using the Perl and then the Perl script with the name run_de analysis.perl, and then we are using the matrix mean minus matrix mean the input file, and then we are using the method minus methods edgeR and then dispersion 0.01. When you just click it here, it will generate a folder named edgeR. There is a folder, and inside the edgeR folder, let's see ahead. First I will run this command, enter. Okay, so this is done. So let's have a look of this folder, differential expression folder ls-lh. So there should be a folder name edgeR. So this is the folder name edgeR. So let's go into it. That is edgeR 6070. So this is the folder. Let's go into it. And okay, so this folder is having four types of the files. So one file is R script, that is that you can use into the R, and then the second folder is this folder, that is edgeR.count matrix, and the third file sorry, not the folder, these are the files, and the third file is this one, that is ending with the name edgeR is a method name dot differential expression and the results. So this is the file that we have generated with this command, and the fourth one is the PDF file of two of its graph. So one is a MA graph and other one is a volcanic graph. So let's go into it and have a little bit details of these results. So first we have to open this file. So copy it and just open it to the editor terminal. You can use anyone, but I'm always using Nano, so just paste it in. Okay, so this is the file that we have just generated. So these were the number of these are the accession numbers of our transcripts. So then this was our Burberry quant, that was the sample A, and the Pashmina was the sample 2, and this column is a log FC, this column is for log CPM, and this column is p values, and this column is for the FDR. So this is the file that we have generated, and now let's I will give you a little bit of the fold change. So this is the fold change. Like if we are considering Burberry and Pashmina, so the fold change is normally you can say there are two expression values, and for example one, like the Burberry is having expression of 50, and the Pashmina having the expression value of 100. So instead of writing both values, we want to write a single value. So that is the ratio of Burberry over Pashmina. So in this case, like this is the value with the 50 and this value is the 100, so that is 1:2. So it's mean that Pashmina having two folds increase expression. So sometimes we are just writing their ratios. So fold change is a ratio of A over B or B over A. So the fold change value is a 23.98. So this transcript is 23.98 fold increase in Burberry as compared to Pashmina. So this is the fold change values. and then this is log CPM value. These are the p-values, significant statistical p-values that was calculated in this step. And sometimes we adjust the p-values and we write another column, that is FDR, false discovery rate. So false discovery rate is also a modified form of the p-values. So this is the file structure that we obtained. So let's see other type of the file, that is a PDF file we have generated. So go back, Control X, so we will be back here. We are back here. So now we are going to look into the PDF file that we have generated. So we will go into the folder on the graphical user interface. So we are in utils. Now we have to go into the Trinity here, then our analysis, then folder is analysis, and then differential expression. And here in differential expression folder we have a folder named HR. If HR, so HR folder, so HR folder, where is the HR folder? So HR folder should be here. So HR folder should be here. So if I'm not wrong, okay, so in differential expression there is a folder HR. Yes, it should be there. So control find, yes, this is a folder. So I have to go here, and these are the 4 files, and I'm going to open this PDF file. Okay, so here we have two graphs. So let's explain these graphs. So let's go 85%. Okay, so this is called MA plot. So M is a fold change on the y-axis, and on the x-axis we have a log counts. So fold change, as I have already explained you, that this is the value of the Burberry over Pashmina, and this is a log value. So definitely Burberry and Pashmina having expression count of each and every transcripts. So whether it is upregulated or the downregulated. So in this case, here is the 0. So all these black region are those genes or the transcripts which are not have differential expression between our two breeds. So our breed, one was Pashmina and other was Burberry, and their phenotypic character was the length of the hairs and the mohair. So all the genes, those who are not having any effect on their expression, they are not upregulated and they are not downregulated. They are having the same expression in both breeds. All those genes are placed here in a black region. So the upper side of this black region are those transcripts, are the genes those who are upregulated. So this is the value that these are the upregulated transcripts. And the x-axis, these are the counts. So the x-axis is the value. Definitely it's a log of mean of normalized expression count of Barbary and Pashmina. So let me give you once again, because these are the expression counts. These are the expression counts of Barbary and Pashmina. We normalized it, and then we will take the mean, and then we will take the log. So that's called log count. So this log count means, let me explain you again on the x-axis. So this is the log value. This is the log of the mean of normalized expression count of Barbary and Pashmina. So this is this is the log count. So all those genes and the transcripts, those who are above this 0 line, especially this red region, definitely there are few genes those who are above the 0 line, but all those are not differentially expressed in both breeds. So these are the few genes, few transcripts. They mention their names, those who are most probably above the 10 log count value. So these are the genes, those who are upregulated. And similarly, these are the genes, are the transcripts, those who are downregulated. So this is what this MA plot is showing us. And there are this is a PDF file. Sometimes Glimma, I think Glimma is one of the R package that you can generate these MA plots. Those are interactive. Interactive means if you will place your cursor on any of the transcript, it will give you the exact values, that what was its count in both of your samples, and in wild types and in other replicates if you have the replicates. So that is an interactive mode. So here in a MA plot, let me give you once again the overview that this is a log FC value, that is definitely a ratio of the Barbary and Pashmina. So instead using two figures, two values, we are just using only one value. Like I have already explained you that if there is a 50 and the 100 values, so it's 1:2. So we can say that in Pashmina, that is 2 folds increase of expression as compared to the Barbary. So this might be the vice versa. So these are the log of the FC values, and this is the log count. Log count means it's the log of the mean of normalized expression count of Barbary and Pashmina. So that's called log count. So the transcripts which are not differentially expressed in both breeds are placed in a black, while these upper region is called are the transcripts which are having up regulated expression, and these are the genes which are having down regulated expressions. So let's go into the next plot. Okay, one more thing here in this plot, the statistically significant, like the p-value, is not counted. So for looking into the plot where we can add statistical significant p on the basis of the statistical significant p-values, we can plot. We have a plot that is called volcanic plot, and that plot is giving us a more real picture that which of the transcripts and the genes having statistically significant. So let's go here. So this is only difference between these two. So again, here is on the x-axis is a log fold change, and on the y-axis there is a false discovery rate, that is the transformed p-values. So we are just plotting our fold change against p-values. So again, here with the 0, this region, the black region, is showing all the transcripts and the genes, those who are not differentially expressed, having the same expression in both breeds. So these are the genes having the same expression level, are not statistically significant differential expression in both breeds. But these red dots, and especially this region where we have taken the accession number of the transcripts as well. So these are the transcripts which are statistically significant upregulation, and their fold change is around 20. And the left side of the graph are the down-regulated, significant, statistically significant down-regulated genes are the expression values here. So you can look here like with fold change 10. So these are the genes like, and then these are the genes with having fold change of 20. So we can say that these are the genes which are upregulated, statistically significantly upregulated in Burberry, and these are downregulated in Burberry. On the other hand, if you are looking for B to A ratio, then we can say that these are the genes which are upregulated, statistically significantly upregulated in Pashmina. This is what we obtained as a result of this step number 6.